This is a presentation by Rebecca Wilkinson, produced in February 2023. The first part of the presentation covers the components of human immunity and non-specific defences against disease. Immunity protects our bodies against infections. Infections occur when microorganisms such as bacteria, viruses, and parasites invade the body. Bacteria can cause illnesses like meningitis and pneumonia. Bacteria enters the body through a cut or sore in the skin. It can appear as a raised itchy bump, developing into a painless sore with a black centre. The individual becomes unwell. The mucosal barriers encapsulate the spores and destroy them. The immune system consists of pathogens and other foreign bodies which trigger allergic reactions leading to skin rashes and asthma. Pathogens cause diseases once they infect the host, replicating themselves and reproducing as a virus, causing illnesses like chickenpox and the common cold, spreading from their host and infecting other organisms. Fungi are eukaryotic organisms that cause surface skin diseases like athlete's foot, a rash caused by a fungus found between the toes that cause dry, white, and cracked skin. It can be transmitted in communal areas like swimming pool, on surfaces that have been previously contaminated, or by touching infected skin. Parasites live in other host organisms and depend on them for survival. They can cause conditions like malaria, which is transmitted by mosquito bites and animal feces. Symptoms are skin bumps, aches, and pains, weight loss, diarrhea, and vomiting. Infections can be transmitted by direct contact, indirect contact, food contamination, and insect bites. Further information on this can be found on this slide. The body has three lines of defense to protect cells and vital organs from infection. The innate non-specific defenses are mechanisms which function to prevent pathogens from entering the body or function to eliminate invaders. The skin is the body's largest organ and most prominent physical barrier of defense, preventing pathogens from entering the body. As long as the skin remains unbroken, it provides an effective barrier for the body to defend against infectious diseases. Further information on this can be found on this slide. The body is able to fight microorganisms using enzymes found in the secretions of internal mucous membranes, which are responsible for filtering infectious agents, pathogens, and allergens, whilst allowing nutrients to pass through. These enzymes consist of multiple layers of epithelial cells, which appear flat and coated with secretions consisting of various molecules and immune cells that fight microorganisms to prevent infectious agents, pathogens, and allergens from invading the body. The digestive tract consists of a series of effective barriers against infection, including stomach acid, pancreatic enzymes, bile and intestinal secretions, including acidic and alkaline secretions, which act to protect the system. Stomach acid, pancreatic acid, bile and intestinal secretions can all be found in the gastrointestinal tract, a pathway by which food enters the body and solid wastes are expelled. The gastrointestinal tract includes the mouth, pharynx, esophagus, stomach, small intestine, large intestine, and anus. It provides a chemical barrier against infection. The pH of stomach acid is generally too low for most bacteria to survive. In the intestine, the pH is much higher but still sufficient to destroy pathogens. The movement of chyme through the digestive system by peristalsis contractions of the intestines helps remove both harmful and dead microorganisms. Pathogens can enter the body through the nasal cavities and airways via the nose and mouth, containing a mucous lining which acts as a barrier between the body and the external environment. The nasal cavities are lined with sticky mucus which attract the microorganisms before they can travel into the airway. An area common to infection in females is the urinary tract. Although it has effective barriers to infection, the female urethra is short and can provide a passage for external bacteria to travel into the body, where it is flushed out of the bladder when it empties. The body's first line of defense consists of specific responses of physical and chemical barriers to prevent entry of pathogens. When an injury occurs, the body creates an inflammatory response involving a number of cellular events. Physical effects include dilation of blood vessels, forming gaps in the cell wall, allowing large blood vessels to pass through, increasing blood flow to the site of injury, creating inflammation. Tissues within the affected area appear red coloration and are warm to touch because of the blood and proteins attracted to the infection, causing swelling and pain. Fever is brought on by the sudden rush of blood to raise core temperature, which has an antibiotic effect that continues until the infection is healed. Inflammation is the body's second line of defense. It consists of non-specific responses and is vital to the immune system, as it isolates the area to prevent the infection from spreading. The inflammation around the point of infection only lasts for as long as the infection exists. Once the infection is over, the skin will return back to normal. As you can see in figure 2, the skin has been damaged and chemical signals have been released from cells to alert phagocytes to move into the area to absorb the bacteria into a vesicle called phagosome. This is known as phagocytosis. Phagosome fuses with mucous membranes containing enzymes called lysosome, which attack bacteria and help protect infection in the process of phagocytosis. The innate leukocytes include phagocytes, macrophages, neutrophils, and dendritic cells, mast cells, eosinophils, and basophils. Macrophages develop from monocytes and form in the response to infection when inflammation occurs. When there is damaged tissue present, these monocytes leave the bloodstream and enter the affected tissue or organ, aiding digestion of large particles in the wound. Macrophages are a variation of cytokine cells, which are small proteins that help enhance immune response, initiating inflammation to attract further phagocytes to the site, producing proteins which block pathogens by attacking the membranes to destroy them. The next section of this presentation covers part 2 of the assignment, Understanding Specific Immunity. Cytokines are complement proteins which are produced by various cells which are used to enhance specific responses which consist of cell-mediated and antibody-mediated immunity. Cytokines activate when triggered by the presence of a pathogen. Antibodies react against antigens which differ from the body's own natural proteins. When two people are exposed to the same pathogens, one might get sick and the other might not. The person who becomes ill has a compromised first line of immunity, either through the skin or damaged mucous membranes. Neutrophils are white blood cells which target bacteria and fungus. They are usually the first cells to arrive at a site of inflammation or infection. The cells attach to the walls of blood vessels and move through the bloodstream to block pathogens from entering through a cut, for example. Monocytes are a type of white blood cell which fight infections and viruses. They are the largest of leukocytes. Basophils secrete chemicals to mediate the body's response to allergies and infectious agents. They light through vessels to allow the flow of blood vessels to the site of infection. They act against foreign invaders, creating 5 different antibodies. They contain histamine and heparin, which act to dilate the blood vessels quickly to allow for the immune cells to reach the infection site. Basophils target infections, recruit immune cells, remove antigens, and complement activation. Descriptions of other immune cells can be found on the slide here. Lymphocytes are a type of white blood cell which are produced from stem cells in the bone marrow that synthesize and express the specific antibodies produced in response to the antigen and later mature and differentiate in the specific organs. There are 2 types of white blood cells, T cells and B cells, which vary in function and structure. T cells mature in the thymus gland and function to help destroy problematic cells such as pathogens. The signal of the immune cells to participate in the immune response, protecting the body from pathogens and cancer cells. Further information about lymphocytes can be found on the slide here. This next part of the presentation covers specific immunity, the third line of defense, self-recognition, and tolerance to tissues and transplanted organs, and immune system disorders. The third line of defense, known as the adaptive immune response, destroys pathogens which have breached barriers of the first line of defense. It also responds to each pathogen and forms immunological memory so cells recognize specific pathogens, allowing the body to respond to future infections. Immunoglobulins are made of 4 chains arranged in Y-shaped configurations, consisting of 2 light chains of immunoglobulin molecules and polypeptide formations, allowing each immunoglobulin class to function in a different way during immune response. During the first exposure of the first pathogen, the acquired immune system learns how to attack and destroy it. Protective response can be made more quickly and with greater force, which allows the body to be protected from harm. The cells of the acquired immune system are mainly the B cells and T cells. Alongside T cells and B cells, there are natural killer cells, but these are part of the innate immune system. B cells and T cells are part of the acquired immune system. Antigens are present on all cells. They are produced by Staphylococcus aureus, which are different to antigens produced by red blood cells. These antigens allow the immune system to distinguish between self and non-self, enabling the non-self to be attacked. Antigens recognize when the immune system encounters a pathogen on the surface of a red blood cell. When an antigen recognizes a pathogen as self, it will not attack it. When an antigen encounters a pathogen it has not seen before, it will be recognized as non-self by the innate immune system. Therefore, B and T cells of the adaptive immune system will respond to the pathogen. Two of the most important factors in allowing the immune system to distinguish between self and non-self are the major histocompatibility complexes MHC1 and MHC2. MHC1 molecules are expressed on the surface of all nucleated cells, acting as an early warning system to the immune system. The cell will degrade these proteins into fragments and display them on the cell surface, allowing them to be recognized as foreign cells of the immune system. MHC2 molecules are present in certain cells of the immune system. They act as an extracellular presentation mechanism, presenting antigens from extracellular pathogens like bacteria. Pathogens are endosensitized by B cells and phagocytes digestive using lysosomes. The antigen fragments are presented by the MHC2 molecules on the surface of the immune cell. This is then picked up by T cells, which target any cell expressing these antigens. Hypersensitivity is an abnormal immune reaction in the form of allergies, not immunity, caused by an immune response to repeated exposure to the same antigen. Autoimmune diseases, for example, are directed against self-antigens, and diseases which result from uncontrolled or excessive responses to foreign antigens. There are 4 different types of hypersensitivity which can be found on this slide. A transplant is a surgical procedure in which an organ, tissue, or group of cells are removed from one person, the donor, and transplanted into another person, the recipient. Transplantation is done to correct the malfunction of an organ system, for example, the kidneys, liver, heart, lung, or pancreas, by replacing the damaged organ with a healthy organ from a donor. Transplantation is often a life-saving procedure. Unfortunately, the immune system does not always accept the donor. This is known as transplant rejection. The immune system is capable of recognizing antigens on the surface of cells as foreign. The immune system protects the body from substances that may be harmful, like cancer cells. During the transplant, the immune system may not recognize antigens present on the organ, and it may initiate an attack. When the recipient's immune system attacks the transplanted organ, it triggers a blood transfusion reaction to match the organ to the recipient to increase the body's chances of accepting the organ. The risk of rejection varies between types of donors, as the differences between donor and recipient can increase the chances of rejection. To improve chances that the body will accept the new organ, doctors use tissue typing, a medicine containing monoclonal antibodies used during and after surgery to fight or specific T cells known as to cause rejection. In order to match both the organ donor and the person who is receiving the organ, the medicine helps suppress the recipient's immune system to prevent it from attacking the newly transplanted organ. The more antigens that are between the recipient and the donor, the less likely the organ is to be rejected. If rejection occurs, it will be one of these 3 types as shown on the slide. Autoimmunity is a system of immune responses of the adaptive immune system to self-antigens that occurs when an organism reacts against its healthy cells and tissues. The mechanisms of self-tolerance fail. A disease which results due to this kind of response is called an autoimmune disease. Specific autoimmune diseases are unknown, as there are many theories about what triggers them, including bacteria, drugs, chemical irritants, and environmental irritants. Diagnosing autoimmune disorders involves identifying the antibodies you are producing. This is done via blood tests and is more accurate than looking at symptoms alone, as many autoimmune disorders don't just attack one part of the body, which makes it difficult to diagnose. Finally, we will cover section 3 of the assignment, showing the biology of vaccination, covering acquired immunity, immunological memory, the benefits of vaccination, and the concept of herd immunity. Active immunity provides resistance to protect the body from a disease or virus such as the common cold, creates antibodies naturally or artificially. Natural active immunity develops from exposure to a specific pathogen, generating an adaptive response. Active immunity is usually classified as natural or acquired. Once the body has been exposed to the pathogen, it creates antibodies to fight the virus, storing in memory B cells, so it can quickly make more antibodies, creating immunity to the virus. Blood types are a form of natural immunity, helping develop the body as a result of exposure to a disease. Further information on natural immunity can be found on the slide. Vaccinations help protect against serious diseases. They are used to initiate the production of memory cells without causing infection. Vaccines contain a fragment of dead or weakened cells, which are versions of DNA of the virus. If the person becomes infected with the virus, the B and T cells will produce antibodies quicker to prevent illness. Newer vaccines contain the blueprint for producing antigens rather than the antigen itself. Regardless of whether the vaccine is made up of the antigen itself or the blueprint, the body will produce a weakened version of the antigen, which will be enough to trigger an immune response. Within the UK, vaccines were introduced with diseases such as smallpox, polio and tetanus. According to the World Health Organization, smallpox had been eradicated in 1980 and Europe had become risk-free from polio in 2002. Before childhood deaths were more common from diseases such as measles, polio and whooping cough, vaccination provides greater protection against serious complications from these infections. In 1974, the expanded programme on immunisation, now the Essential Programme on Immunisation, is established by the World Health Organization to develop immunisation programs throughout the world. Live vaccines are compromised with viruses and bacteria that have a weakened immune response. When live vaccines use a small dose of the virus or bacteria, the body replicates to stimulate an immune response before it runs into an active case of the same disease within the body. Inactivated vaccines require multiple doses at the first dose and do not give immunity, but do prime the second dose. The immune system stores this information known as immunological memory. By examining measles infection rates in the UK between 1940 and the present day, in order to assess the advantages and disadvantages of vaccination, measles infections were widespread in the UK prior to the introduction of the vaccine in 1968. However, measles infection rates have progressively decreased since the introduction of the vaccination. Vaccination is an effective way to protect the body from infection and disease, and the data on measles infection rates. Measles is a highly infectious viral disease which can lead to serious complications such as pneumonia. The measles virus is highly contagious and remains a leading cause for infant mortality. The Health Protection Agency website has a page listing on the number of cases of measles and the number of deaths from the disease in the UK from 1940 to the present day. 10 years prior to the introduction of the single measles vaccine from 1958 to 1967, there were 863 deaths and 4,120,936 cases in the UK. In the period that people were vaccinated from 1968 to 1977, there were 292 deaths, a fall by 66%. The measles vaccine prevented millions from becoming infected. From 1940 to 1968, there were 7,863 deaths. After the introduction of the measles vaccine, deaths reduced from 85 per year to 20 per year. 20 years later, figures fell to just 1.5 deaths per year. Between 2001 and 2013, there were a rise in the number of UK measles cases. There were 3 deaths. In 2018, there were 966 measles cases, which then dropped to 259 cases in 2017. It is important to get vaccinated to create antibodies against diseases like measles, mumps and rubella to enable the immune system to prevent the body of future attacks from diseases, helping to protect us over a lifetime. The MMR vaccine was created for measles, mumps and rubella, as they are highly infectious conditions which spread easily among some vaccinated adults and children. Getting vaccinated is important as these conditions can lead to serious problems including meningitis, hearing loss and problems during pregnancy. 2 doses of the MMR vaccine will provide the best protection against measles, mumps and rubella. Further information about measles can be found on this slide. Access to healthcare and public education plays a role in determining vaccine uptake or the percentage of vaccinated individuals. Vaccination programs ensure that the population is adequately protected against disease. Vaccination programs and vaccine uptake are important components of the body's acquired immunity. They are important for achieving herd immunity as they help increase the number of vaccinated individuals. Herd immunity helps to lower the infection rate among unimmunized individuals for infections that are transmitted from person to person, such as influenza. Not everyone needs to be immunized to control the disease. However, for individuals who reject vaccination, it will be less effective for them as they will not be protected. High vaccine coverage rates of influenza immunization in healthcare, for example in care homes, can result in lower patient mortality in influenza-like illnesses. Some healthcare workers remain unvaccinated because they do not perceive influenza infection to be disabling and life-threatening. Instead, they are concerned about the side effects of the vaccination.